why I wanted you to see this. We're going to talk about some lining cells. So when we're done, we're going to look at some cells. Because body fluids, you got to start not only knowing your normal white cells that you've already done in hematology, but you're going to be expected to know a lining cell versus a macrophage when you're counting your diff. So I get real diffs from the hospitals so that you can see lining cells. But one of the lining cells in spinal fluid is called choroid plexus cells. And sometimes you'll hear them called ependymal cells. You just have to know it's a lining cell and it doesn't mean anything bad for the person. Just like mucus doesn't mean anything bad, squamous epithelial cells don't mean anything bad. Think of that way with lining cells. They're going to be present many times if they're going to do an invasive procedure through a sac or through an area. So that's the one thing. The other thing we're going to talk about is subarachnoid hemorrhage. When you get these 3 tubes, if there's blood in every tube that looks the same, they're going to worry that they have a hemorrhage in the subarachnoid area. If you only have blood in the first tube and it gets less in the second and third, they're going to say it's a traumatic tap. They got blood from the invasiveness of going into that space to get the spinal fluid. So those are terms you want to be aware of. Subarachnoid hemorrhage, and one of the lining cell names is called choroid plexus. There's also one called ependymal. But they're one and the same. So on a test, if I show you a cell of spinal fluid, that's a lining cell. You can't say lining cell. You have to say choroid plexus or ependymal lining cell. Because when we get to serous fluids, they're called mesothelial cells. But they all look the same. Your lining cells look the same. And they're very, very big. If you know monocyte is your biggest normal blood cell, these are twice as big. You might get a few lining cells that are smaller, but overall, they're at easily 2 to 3 times bigger than your biggest white cell, which is your monocyte. So we know the fluid protects the brain, lubricates the CNS, carries nutrients, is an excretory channel for waste products. Normally adults have 90 to 150 mils, newborns 10 to 60. We get a lot of newborn babies with unexplained fevers, maybe a blood infection, and you're going to be lucky to get adequate amounts in those 3 tubes, and we're lucky to get a few mils in each of those 3 tubes. So this is the volume they have. We're lucky to get, when they take some, a couple mils per tube. So it is our biggest indicator of bacterial and viral meningitis. Probably that's your 90% of why you're going to get one ordered in the regular lab, even though you might be able to, like a spinal fluid could detect MS. You could do protein electrophoresis, and you're going to get these things called oligoclonal bands that are classic for MS, and they can do it on spinal fluid. Probably your career in a normal hospital lab is going to be to rule out viral or bacterial meningitis, your most common reasons. Encephalitis can be found, MS. When I worked in serology, Diane's going to teach you RPR, or syphilis testing on blood. We also did it on spinal fluid, and they call it neurosyphilis. If someone has syphilis that goes untreated, and it's very easy to treat, you just treat it still with penicillin, it can develop neurosyphilis. So you're going to get a lecture on that, the stages of syphilis, and so we always did spinal fluid testing for syphilis. And the subarachnoid hemorrhage, you're going to be able to see that just on how your 3 tubes look when you get them delivered to the lab. So on our test, this is for put test 5 next week. It's not this week. So test 5, you're going to be able to tell me how can you differentiate a subarachnoid hemorrhage and how can you differentiate a traumatic tap. You gotta know they're 5 ml tubes, and like I said, you're lucky if it's a good puncture to get 2 mils in each tube. Sometimes the puncture is great and they can get full 5 mils in each tube. You have to know who gets it first. Chemistry gets tube 1 because it's not going to be impacted if there's some blood from the traumatic tap. Plus, what if they don't clean the skin well enough? Do you want to go into microbiology, the first tube, when you're worried if they have bacterial meningitis and you might have bacteria from the skin because you didn't clean it well enough, or whoever was doing the cleaning before the puncture? So it's a total protein and glucose, and we're going to go over how those values help you. So you're going to be able to tell me what's the difference of bacterial versus viral. Tube number 2 goes to microbiology. They're going to do a gram stain stat. You can't culture it stat, but you're going to do a gram stain stat. At St. E's, we had to filter our crystal violet. That's your primary stain when you're doing a gram stain. We had to do a filter to collect any sediment that might be in our bottle so that only stain got on it, because we don't want to call gram-positive cocci as a stat gram stain when it could be sediment because you didn't filter your gram stain. So we always took the extra precaution, filtered our crystal violet when we did our gram stain. And the culture, we know cultures take, well, you probably don't know, cultures take minimum 48 hours. And if it is anything that could be an anaerobe that grows without oxygen, you're going to hold that culture for like 5 days. So it's not fast. So the gram stain, if there's nothing on it, you're going to feel that you probably don't have a bacterial meningitis. Then the last tube is hematology. It does matter if that's true blood there from a subarachnoid hemorrhage, or was it a traumatic tap. You want to go the furthest away from the tap, which is the 3rd tube, and normally that's the main thing they collect. So you got to know for your test what tubes go where and why. Why would you not do micro or hematology test on tube 1? The blood or the bacteria from the skin. So if you understand why you're doing the order, it'll help you to remember it a little better. But that's a big deal. What order where it goes to. And we're happy if they have a 4th tube if you have additional testing. Sometimes they want to do fungal testing. They might want to see if you're someone who has a fungus called Cryptococcus neoformans. You're going to all get to do a serology test in lab at your clinical called a cryptococcal antigen. Remember we talked a little bit about antigen antibodies? Blood ones are natural. Well, anything foreign is an antigen. Whether it's a fungus, whether it's a bacteria, whether it's a parasite, your body sees that as foreign. So if they detect cryptococcal antigen in whatever fluid they're collecting, and it's positive, that tells them you actually have the fungus in that fluid, whether it's a blood or a spinal fluid. And then the test kit, if they're looking for cryptococcal antigen in your fluid, let's say blood, the test kit has to provide the antibody to that antigen. That's serology. Remember, antibody, antigen, lock and key, they fit. And a lot of departments have serology tests, even though they might be micro, or they might be chemistry, or they might be serology. You're going to do color and appearance just like a urine. The typical appearance of a spinal fluid, it looks like water, clear colorless. And our rule of thumb was, you're going to count all 9 squares, so you're going to do the whole grid. It's going to be straight plated when it's clear colorless, no dilution. But if you even saw 1 white cell, you have to do a differential. And anybody remember from the last PowerPoint, which is on test 3, how do you do a diff that makes it doable, that you can count pretty quickly because of a process? It's a centrifuge, and remember the name of it? It's called the cytocentrifuge. It's on your last week's PowerPoint, and it puts all the cells, you put a piece of filter paper that has a hole in it, and it's in the centrifuge, all the cells that are potentially in that fluid go right in that little circle. So you go do a diff, and you're reading one little area, and some of them you can read in 2 areas and you have your 100 cells if they're cellular. So if it's clear colorless, it's tough to do a diff, but our rule of thumb was at St. E's, anytime you even saw you had a manual count of 1 white cell, you were doing a diff. Even if it was 1 lymphocyte, then you reported 100% lymphs, because they knew it was clear colorless, you hardly had any. That was our rule of thumb. If it did not have any white cells on the manual count, then you didn't have to do the diff. And they would see, well, there were no white cells, what am I going to do a diff on if there's no white cells? Red cells, you're almost always going to have something. Ironically, newborns allow up to 30 white cells and consider it normal. So I don't know what it is with their spinal fluid that's different from an adult. Probably adults are up to like 5 that they don't worry about. Naturally, just like a urine, cloudy, even hazy on a spinal fluid, you're going to worry about. But it can typically be cells from just the tap, okay? And then bloody, you only worry about blood if it's even distribution. If it's the first tube has more than the last, then it's probably a traumatic tap. Sometimes you'll even see clots. If blood clots form, you might on a traumatic tap. This is a big thing, you want to know that I have bolded. When you centrifuge, you will centrifuge for chemistry. You will centrifuge your spinal fluids before you run your glucose and your protein. If it is a traumatic tap, you're going to have colorless, just like when we spin down our cold agglutinin and to wash our cells, and it's colorless when there's nothing bad going on, it's just red cells, and then you wash them when they're fresh. Same thing with when you spin a fluid that might look bloody from a traumatic tap. It should spin down where the supernatant, what's on top of the cells, is colorless. Subarachnoid hemorrhage, they call the term xanthochromia. It could give a yellowish color, a pinkish color to the supernatant. So anytime, if they put on a test question on the registry that it talks about a pink supernatant, a yellow, or they use the term a xanthochromic supernatant, you should be thinking subarachnoid hemorrhage. That there's been bleeding into the brain, that is old blood that breaks down to hemoglobin, and that's going to be. But that term goes with spinal fluids, xanthochromic. And it usually goes hand in hand with a subarachnoid hemorrhage. You don't see that with a traumatic tap. I don't know if it's very easy to see. We can see, if you can tell the ones, if you turn the lights off. Let's see, and that part didn't help either. So the first one, A, is normal spinal fluid in the 3 tubes. And then B is supposed to be a CSF from a fresh hemorrhage. Do you see it's the same in all 3? So you're not going to call that a traumatic tap because it's the same in all 3. You're going to worry that they're hemorrhaging into the brain. Then the third one is supposed to be xanthochromic CSF from an old hemorrhage. It has a yellowish color, if you can tell the yellow compared to the clear colorless. And then a traumatic tap, do you see how tube 1 is so much bloodier than tube 2 is better, and by tube 3 it looks fine. So you guys have to be able to know, even when you're working, when you might worry about a subarachnoid hemorrhage. So that's just your first clue. Even distribution, there's going to be some cells we'll go over that you'll see in a spinal fluid that also tell you that you could have a subarachnoid hemorrhage. That's the pinkish color. So either pink or yellow color when you spin them down, and the primary area you're going to spin them down is when you're doing your protein and your glucose. Because there's minimal hospitals that do bacterial antigen. I loved it. The one time, I've only had 2 bacterial meningitis in my career. One was a Klebsiella, no it wasn't. It was Haemophilus influenza, which is going to be in micro, a very sharp little gram-negative rod. And then my other one was a strep pneumo on an elderly patient who had an ear infection and went to the dentist on Friday, and by Saturday he was in the ER with a meningitis. And in the Haemophilus influenza case, the bacterial antigen panel helped because it told us. It was one of the ones you tested for, and he was positive for strep pneumo. So I personally loved having the bacterial antigen panel and not just the gram stain. So Gateway, where Katie's going, she'll get to, if they get a spinal fluid, they're going to have the bacterial antigen panel. And this one is you don't see as much on the traumatic tap. You don't see as, I don't know if they even have 3 tubes. They just have this one, the traumatic tap. This one, I'd rather it, for me, I would give you something that showed more bloody than this. This almost looks like the 3rd tube of that last picture instead of the 1st tube. But do you see even distribution on subarachnoid hemorrhage? So that's the big key that you want it. There should be less blood in 2 and 3 on a traumatic tap. So for kids, for some reason, children can have 0 to 30 white cells and considered normal. On red cells, 0 to 5. On adults, 0 to 5, and no red cells. But most of the time, if you see the tubes and there's more blood in tube 1 than the other 2, you know what blood's present is going to be because of the tap. And the other big thing is key, and you should know this from hematology. On a bacterial infection, what do you expect to see high on your differential? Neutrophils. And you're also going to see a shift to what? What the term? Shift to the left. So you might see bands, you might see metas, you might see myelocytes. So if they say a shift to the left, you remember that's a big thing. Viral meningitis, you're going to have increased lymphocytes. We as adults, our normal white cell we have the most is a lymphocyte. Kids, the most common white cell for them are monocytes. So lymphocytes, we tend to have more lymphs normally. Newborns tend to have more monocytes. Neutrophils, bacterial meningitis, any kind of bacterial infection, CNS hemorrhage, lymphocytes. People with viral meningitis is your number 1. Bacterial meningitis, the first ones are your key ones you're going to see. People with parasitic infections, I don't know if there's any one that would be common, but you'd probably have to been exposed at an area that maybe they have poor water and sanitation. When we get into parasites, a lot of the people that get them are areas that have poor water, poor sanitation. And MS can tend to have more lymphs, and they can see more monos. You always associate EOs with parasitic or allergies, but they can be elevated also if you have a fungal spinal fluid infection. And then we're going to look at pictures that show a siderocyte. You're going to have to look at 3 cells that go with subarachnoid hemorrhage. One will be the hematoidin or the hematin crystals that are in a macrophage. One will be a siderophage, which looks like black iron from red cells is speckled all over. And then another one is a macrophage that has ingested red cells. When you have a bleed in your brain and it sits around, you end up getting these cells that can show in your differential. So for me, you're going to have to tell me what are the things associated with subarachnoid hemorrhage. You already know the 3 tubes are even distribution. They might have a siderophage, which has iron ingested from the breakdown of red cells. It might have iron broke down, and there's these hematoidin crystals that show up in the macrophage I'm going to show you. And then they might have a macrophage that's gobbled up red cells. And you'll actually see, like a little Pac-Man, the red cells ingested. So when we go over the slides at the end, you're going to see that. And then naturally, you always worry about malignancies. St. Elizabeth's, we couldn't call anything. We couldn't even call a mesothelial cell or a macrophage. We had to say, when we were doing our 100 count diff, large, unidentified mononuclear cells. Okay? And you don't differentiate your neutrophils either. You just say it's a neutrophil no matter if it's a seg, a band, whatever. You say it's a lymph or you say it's a mono. Those are your three common ones. But if it's a malignant cell, for me, you're going to have to say, yes, that's a mesothelial, because I'm going to make you identify lining cells or a lining cell. You're going to have to tell me if it was a macrophage, and you're at least going to have to tell me if it was an immature or malignant cell. Where you work will allow you whatever they allow their techs to call. Then we had to put in a code, see pathology report to follow. So don't feel that you're going, oh my gosh, I'm going to call what malignancy that is. No, that's going to be the pathologist's job. But you don't want to miss a malignant cell to at least say, hey, you want to look at this immature cell. And the other thing, get in the habit, where do we read your diffs? At 100. Get in the habit of scanning it first. So you got this little hole that all the cells are pushed in. Don't just say, you have let's say it's very cellular. You count it in 2 fills and I'm done. Scan it first. Make sure you're not missing an area that might have immature cells before you start counting. That is a good habit to get in. I would say serous, the most common fluid I did was pleural, which is lung, and lung carcinomas can be pretty typical out there. And I'm going to teach you, and when we get to serous, how to distinguish whether it's a malignancy or a chronic body condition. So the predominant cells, I put in a little chart. I think this is from Strassenger. So this kind of goes through all the cell type, the significance, and microscopic findings. So I put that in your PowerPoint for you. And then differential diagnosis on all types. We're going to key into bacterial and viral. Our probably 90% of your career on a spinal fluid, and those are the two things we're going to worry about the most common things. The big thing we're going to see with bacteria and viral, they're both going to have elevated white counts. We already know neutrophils should be the dominant one in bacteria, lymphocytes the dominant one in viral. The protein is going to be elevated in both. It says it's more elevated in bacterial. For me, you just have to know the proteins you get from chemistry are going to be elevated in both. This is the big deal. Glucose is markedly decreased. And usually it's probably about 75% of your normal glucose. So you're looking at 45 to 75, whereas your blood is what, 70 to 110, 60 to 100, depending on what lab you work in. This is something I'd circle. In viral, the glucose is not eaten up and used. Remember, what did we say in urines that sometimes the bacteria proliferate in someone who has a diabetic condition because the glucose is in the urine and they keep growing. Well, in this case, they eat it up from the fluid and it's decreased. So that's a big deal. So far, neutrophils is a big way to distinguish it, and your glucose level is decreased in bacterial. Okay? And then I don't know if you're going to see any labs commonly do a lactate level. If they do, this is another way to distinguish. It's very elevated, greater than 35, and it's normal. I don't know if you're going to see that done in the regular labs. Glucose and total protein are commonly the two chemistry tests they do. And naturally, you're going to have a gram stain that shows if it's a strep pneumo, I'm going to have gram-positive cocci, or they'll be in pairs. We call them diplococci. If it's a Haemophilus influenza, do you know until they did the vaccination called Hib, usually by the time they're 2, that was a very common meningitis, was Haemophilus influenza, and it's not gram-positive. They're pink little bitty tiny rods, and That's why I like the bacterial antigen test. We could test for strep pneumo, Haemophilus influenza, group B strep, because a lot of women carry group B streptococcus in their vaginal area. So sometimes on delivery, they will give that group B strep to that baby. And then Neisseria meningitidis, there's like 13 serotypes. We had 2 antigens for Neisseria, and one of them was linked to E. coli as well. So it was a great test to me. You did it immediately, had feedback if there was one of those present because it clumped. Kind of like we did red cell clumps, these were white cell, not white cell clumps, but clumps of white specks. So these are the 2 columns you want to star that you're going to have to know for me for test 5. Bacterial versus viral. That's going to be your key thing you're doing in your the real lab. And we already, usually the protein is 8 to 32 as a normal range. Bacterial infections is the most common one. The bacterial meningitis, you're going to see it. But it can be also elevated when there's red blood cells involved with hemorrhage and traumatic tap, which makes sense. Anything that is made up of protein. If you're leaking fluid or fluid loss, you might have decreased levels. And I do want you to know the term that distinguishes MS on a spinal fluid. If the doctor suspects MS, they will order a protein electrophoresis to see if they have these special bands. Yes?
The 2 charts that you showed us, is that graft or structure?
Let me look at, I can tell by the table. I think they're usually Strassenger. Let me look through the chapter. No, maybe not. Let me look at this one. No, this is Strassenger here. Yeah, it looks like there's Strassenger.
Okay.
Yeah, and there's a Strassenger book in there too. So there are, like I said, Graf's pictures are better, but I think Strassenger is a little bit more, I don't know if you can see it, but on a protein electrophoresis, on someone that has, this is normal for CSF, and a person with MS would see this intense staining in this region. So just so you know the term, that oligoclonal, that's one of those terms, if you at least know the term, you'll get it right with MS, even though you may never get to do a protein electrophoresis in your life. Back in the day when I was in chemistry in 81, 80, they had special electrophoresis, and these are actually pretty fun procedures because we did CK enzymes, isoenzymes. Did you at least learn them with CK? You know, the 3, and LD, the 5. We did CK isoenzymes, LD isoenzymes that would determine heart attacks, liver problems, whatever. And we did protein electrophoresis, and you guys had to identify the 5 areas, albumin, FL1, with Diane will go over gamma, and then you'll get to see how you determine a multiple myeloma or macroglobulinemia. Well, I don't know, there's not much electrophoresis being done except protein and this thing called an IFE. But it was actually a pretty neat little department called special chemistry. Okay, the glucose around 40 to 70, usually there's 60-80% of the blood level. And you might see someone in a diabetic coma, they've noticed they have increased glucose levels with their high glucose. But the big one is the decrease, this here I would highlight. Decreased in bacterial, normal in viral. The viruses do nothing with the glucose that's present, whereas the bacteria use it to multiply. Lactate, the normal is 11 to 22. I think on that chart it said bacteria was greater than 35 and it was normal on the viral. I don't really know if you'll get to see that test done. I think most labs just do glucose and protein. And then this is just a little synopsis of I don't know anybody that does glutamine that's in a normal lab, maybe a reference lab. It says if it's greater than 35, you might have some disturbance of consciousness. I don't really, these two, I think you'll be lucky to see them at like a Quest or a reference lab that someone maybe wanted them to send out. This is going to be your 2 common chemistry tests ordered at a real lab. And you know, we're going to do discussion board when we're on at clinical. And what I asked the students, it's pretty good. It's going to be under MLT 245, which I hope everybody's signed up for that. You can't go to clinicals without being signed up for it because our liability insurance kicks in. No one can be at a clinical without being in that class because that's how you're covered. Anyway, we do discussion board, and one of the things I ask the students to, every week there'll be 2 points extra credit questions if you choose to do it. If you choose not to do it, you just don't get any extra credit points towards your clinical grade. But what I have the students do, I post a question that I want you to answer it involving what you're doing at lab. But I also like when you respond to each other. Last year's class wasn't good to respond to each other. They'll just post something. Where they would post good stuff, but they wouldn't like say someone was telling about, like, let's say I'm out and they do a lactate test. Say, hey, here at Children's, we do lactate levels on the spinal fluids on the kids. And someone might say, post to that person and say, oh, we do too. But no one did that. They would just post their little answer, and then I reply to everybody, okay? But I would want you to try to use it more interactive, that if you see someone's post that hits home with what you're doing, reply to them and not just put up a post and then I reply. But it's kind of a way to stay connected. It's been a really good thing. And then I do it for at least 6 of the 8 to 9 weeks, depending on which clinical, and you get 2 points per post towards extra credit on your grade. And so one of the things I asked, and Deanna got a job in micro at St. Anthony's, and they do a lot of molecular there before this thing called MALDI-TOF is coming into place. So I asked her to come back and give a lecture in January in our class because this Maria, her head, was telling me all the molecular they want to add. So I want to meet with this Maria before I do update my syllabus and have her add the molecular tests they do to our lines that they check off on. And Deanna is a whiz with the computer, so I said, hey, would you want to come back and give a lecture on your molecular test that you're really doing because you're working in micro? So she's supposed to come back sometime in your micro class and give like an hour lecture on what she's doing because she's in micro at St. Anthony's. And so use discussion board to enhance everybody's knowledge, because you might get to do something neat that we didn't talk about or in any of our classes that they're doing that's neat. So anytime even if it's not a question I directly asked, if there's something good you do, talk about it on discussion board, maybe even the principle of it or the background. You're going to have to know these, and these are the hard ones because you haven't had micro yet. You've got to know what is on a bacterial antigen panel. Staph aureus is not included on the panel, but it is actually an organism that could possibly cause the bacterial. None of the panels that are out there include staph. But staph is one of the easiest bacteria to identify in the lab, even day one. They have this chrome agar. They can grow it, and the next day, if it turns this agar pink or blue, whichever kit they get, you have Staph aureus. Then they just got to do a test to make sure if it's resistant one, called MRSA. So you've got to know these 5. You've got to know there's 2 streps, so if it helps you to write them in an order that might help you to remember them. But you're going to have at least a matching question, and you got to be able to detect which one's not right. So we'll call it group B, and I'll just say strep. Its real name is Streptococcus. We always call it group A strep, group B strep, that kind of thing. So there's 2 streps. Then the one little kids hardly get anymore because they have the vaccination is Haemophilus influenza. and there's many serotypes, and it's serotype type B, so they call it Hib, Haemophilus influenza type B. Then there's 2 different sets of Neisseria. Neisseria has like 13 serogroups, but we need to know meningitis. There's one called A, one called C, and one called Y. On the test kit, it'll be a little vial, and it says Neisseria ACY, so it would detect all 3 of those serogroups, A, C, or Y. And then this one was the hard one, and I'll give you an example of why it was hard depending on who your patient was. This is meningitis. I'm just going to put a period. It's that same name. B, but it also could test for E. coli. So it had 2 unrelated antibodies, and these were what the patient had in their body fluid. Spinal fluid, blood, urine. So the antigen in patient body fluid. The kit had to provide the antibodies to these. So just like we talked in serology, so this is a test on a spinal fluid, but it's still a serology premise. So anytime I'm looking to see if I have the actual live microorganism, I am looking for the antigen in the patient's fluid. Then think, the kit has to provide the antibody. So you got to know what you're testing for. Like when we did the cold, we were looking for the antibody in the patient's serum. So the kit, which our kit was the red cells, provided the antigen. You've got a serology, put in antigen antibody context. So on your test, you've got to know these to at least recognize one that's not in there. Staph aureus is a culprit for meningitis, but it's not on the panels. It's not a panel organism. There's a stain we'll talk about in micro for tuberculosis called acid fast. Did you talk about that at all if you've had the anatomy here? So tell me, does anybody know the difference of what the primary stain is in a gram stain versus an acid fast stain? If you had that in micro? Nope.
There's a red one.
It's a red one, that's
I
good.
don't remember that.
That's good, because the crystal violet is purple. For a gram stain, your primary stain is crystal violet. On an acid fast stain, your primary stain is called carbolfuchsin. The counter stain is methylene blue for acid fast. When you counter stain a gram stain, you use a red stain called safranin. So you're going to have to know those. So if they suspect TB, it's not just going to be done if you don't tell them, hey, we suspect this person has tuberculosis, can you do an acid fast stain? They have to order that. That's just not going to be a common thing. They're just going to do a gram stain. And it's just a gram-positive bacilli on a regular gram stain. And this, kind of like we said with oligoclonal bands, if you know India ink, it's always a stain for a capsule on a fungus called Cryptococcus neoformans. Most of you, if you get to do anything with it, are going to do a serology kit for a cryptococcal antigen. But for the registry, you're going to know India ink is a stain they can use for Cryptococcus neoformans. I think all of you will do a serology kit, but that's something you need to know for your registry. And we'll see that too when we go through slides. And then this is just different causes of different diseases. Pleocytosis is just cellular increased neutrophils, pleocytosis of lymphocytes, pleocytosis of eos, monos. So that was one, I'm sure it's from Strassenger that I put in there for you. Okay, India ink, and then we'll see when we go to the slides too. India ink that has a capsule. So what they do is they try to use a stain that shows the capsule. And they have to suspect it and order it. So they're not going to just do this on every spinal fluid. The physician has to suspect that you have fungal meningitis. But India ink is the stain they use. Just like acid fast stain is something they use for if they suspect TB, the doctor has to suspect that. It's not going to be on your normal spinal fluid. They have to, the special things for fungus, the special things for TB, they have to suspect. Okay, so we're going to end with going through some slides, and we'll do this, yes.
So cryptococcus, that is.
Fungus, Cryptococcus neoformans is the fungus that commonly gets asked about on the registry. It's called, and in micro, usually the genus is capitalized and the species is lowercase. It's something you're just going to have to get used to. Micro is like a different language, okay? Most of you are going to do a serology test kit called the cryptococcal antigen. So we're looking for the cryptococcus in the body fluid. So it's the antigen the patient provides. The kit has to provide the antibody if you have the antigen. If you don't have the bacteria or the antigen for the fungus, or if it was a TB test that looked for an antigen, then it's not going to react and it'll be completely negative. So remember, anytime you're looking for these in a body fluid, the patient provides the antigen. It is the infective agent. Yes?
I forgot.
Well, I mean, we're going to go through some slides, so think about it. So first, what are our slides for this test on Wednesday?
Counts.
What did I say would be the slides? 10 of 12 of what?
That wasn't last test.
Cast and all crystals cast.
That's part of this test coming up?
We're doing those first. We're doing what you're going to be part of your test, and I'll do the same thing. If we have time tomorrow, I'll give you the test, and if you're satisfied, you don't have to take it Wednesday, and you don't have to get here till 8:10. So we're going to handle it the same way. If you don't, and we'll do it right away, right at the beginning. So if you're late or whatever, you miss it, then you just take it the real day. And on the real day, we'll do it right at 8. If you're late, you miss it, you just jump in.
This one we going on now is for test wings.
Yep, because these are still urine, but I wanted to go through the cast and the
So it's only going to be cast and crystals?
cast and crystals. I'll give you 12 and we'll do it the same way, the best 10 out of 12. So you know to review your cast and crystals, okay?
This one is 3, right? You're in 73.
How are you going to give
Well, it's in all of them.
Oh.
I'm just showing them my latest one because I put 3 pictures per slide. You can look at urine sediment 1, 2, and 3 to review, but urine sediment 3, I posted 3 pictures per slide. That's why it's easiest for me to go back and review it, because then you can look at 1 and 2 yourself. And if you want more, just do Google images and put in what image you want. You know, if you're sitting, and the best thing is to be on the computer anyway and look at them. So just take the time to get on a computer, and then if you want more, just do that. So remember, the Hyalin, depending on phase, anybody's going to love having phase when it doesn't take color, even if you use stain. Okay? But the bright field, you still have to be able to lower your light. But I think you guys have done really well at finding the Hyalins. And we have only bright field. So you have been trying to adjust your light so you don't miss them. And remember, what is the other key about not missing them? Where are you going to scan if you're doing them like we are doing them? The edges. Put that cover slip on and they tend to go to the outside. And what's going to be your clue on the dipstick? High protein. Okay? Goes with cells, cast, bacteria. You get high protein, you better scan the edges. Okay? Hyalin is the base, yes.
With the crystals, are you going to use the pH
Yep.
pictures? Okay.
Yep. You will get the pH, especially On your pathological, I'm still gonna say they're acid, they're all acid. But on your normal ones, you know you're not gonna call ammonium biurate if the pH is less than 7. That is your biggest clue, is your pH when you're calling a normal. So it's up to you to study which ones are normal acid, which ones are normal alkaline. Your red cell cast, I will, and once again, I'll let you look at them on the computer screen or up here. I won't just make you get them from a picture on a black and white paper. So you'll be able to look at the computer or look up here, and I'll try to find casts that have cells that help you. These all have cells, so when you're working, remember, you don't want to be calling a cast of a cell that you don't see any free. You don't call any renal epis, and you call a renal epi cast, someone's going to come looking for you. What is this? This does not match. This does not go together. Now, reds are different because reds, remember, you can have large blood, and if it's all broken down to hemoglobin, you're not going to see any red cells. You might have 0 to 2. So if you have a cast, they might all be broken down, and it could be more of a hemoglobin cast, and you will see, usually it takes on the reddish brown look though. Even when you're looking in a phase with or without unstained. But what's going to be your biggest clue on the dipstick? Blood. You don't call a red cell cast or a hemoglobin cast if you don't have blood flagged. Even if you don't see them microscopically, you better have it flagged on your dipstick. That, even the ones I've been bringing, we've been able to see white cells, even if it's a few hours old or the next day, but we've hardly been able to see any red cells. They break down quickly. Your white cell cast, I'll try to find pictures that at least give you white cells in the area. What is the clue that it's a white cell versus a renal epi? What kind of nucleus should you see if it's a renal epi cast? It should be a singular nucleus. Usually the white cells just look granular, and they're neutrophils, so they're not going to be a single nucleus. And they're going to be smaller than a renal epi. But if you see a cast that has single nuclei, think about it being a renal epi. But what should you have flagged? You better have called some renal epis. Don't ever call a cellular cast without either having blood on your dipstick for blood or hemoglobin. White cells, you better call some white cells. Renal epis, you better call some renal epis. This one's pretty good. They look like single nuclei in a lot of them. And they look actually bigger. Now, what do you think this is? Yeah, budding yeast. This one, there's some good single nuclei as well. They just do not look like granular white cells. And look how much smaller this is to even this size. And then these have more single nuclei. So that's how we'll kind of distinguish. But bottom line, don't call a cellular cast unless you have blood on your dipstick for a red cell, white cells for a white cell cast, renal epis on a renal epi cast. And in the progression, your cells then break into a granular cast. The granular cast, if there's no voiding and there's the stasis, they're not voiding, then they become the waxy or broad. And I don't care, for me, you can call it a granular cast. You can say, oh, that's a fine granular, or that's a coarse. This is probably the finest one that I would probably call, of the 4 up there, a fine granular cast. These to me are all more granular. Remember, and let's say you can see a cell down at the bottom. The bulk of it is broken down into granules. Go with a granular cast. Do whatever is the most accurate. If 75% of the cast looks granular, go with granular. And then the waxy or broad, I don't care, but remember, you still want to put cast and crystal at the end of every name, or it's a half point off. Now, and the red cells we won't. So let's go back to normal. Okay, so the acid ones would be calcium oxalate. uric acid, and any amorphous, you just call it amorphous sediment. I'm not gonna make you distinguish urates or phosphates depending on the pH. Okay? The clue for urates is the pink sediment. But you can still have amorphous above, they call them phosphates when it's above 7. For me, you just wanna say amorphous sediment. I think most of your labs, since you get very few alkaline urines, most of them are less than 7, most of you are gonna be fine with just calling it amorphous sediment. None of these are bad for you. So all of them could be in all these shapes. The most common one we've seen, and this looks like little bacteria, which would be many in the background. This is kind of hard to distinguish if it's amorphous back here. It's not as clear cut, but it looks like it might be bacteria. And this one, so remember, calcium oxalate has other forms. I'm only going to test you on the most common form. And I think that's mainly what we've seen in lab, is the star or the X in it. And then uric acid is the one that can be varied. So I think all of you are okay with amorphous sediment, it seems, when we're in class. And I think you're all okay with calcium oxalates. Remember, uric acid is the one acid crystal that's normal that varies in size and shape. Calcium oxalates are usually the same, and the uric acids vary. So if you see it's an acid pH and they vary in shape, then you should be thinking uric acid. I think we even had a little barrel shaped the other day that looked almost like a little barrel. I would say this is probably a very, very typical shape, whether it's turned straight or at an angle. I would say that's probably your most typical shape of uric acid. But remember, use your pH. They're not anything bad for the patient. Just some of us have stuff that precipitates out. Your amorphous sediment, that's all you got to call it for me, is amorphous sediment. You don't have to say urates or phosphates. Now the pH greater than 7, triple phosphate, remember if they're flat, they look more like an envelope. If they would be turned to their side, they kind of look like a coffin lid. Most of these look like envelope. These might be a little turned on their side. That one might be a little turned on its side. But most of the ones, the flat are the envelope shape. Calcium carbonate are the dumbbell shaped. And these two, don't they almost look like leucine? I mean, they almost look like cut through a tree trunk but the dumbbell shaped, and what's your big clue you're going to look at before you, when you set, is the pH. It's not going to be calcium oxalate or uric acid if the pH is above 7. And then the thorny apple, the ammonium biurate, I call them the little aliens. And then pathological, they're all less than 7. These are the 5 you have to be able to know. Cysteine, and what amino acid aduria did it go with? Because that's on your test too, which we'll review them tomorrow. The one that has the kidney problem, not an amino acid genetic problem. So it's cystinuria, the kidney has the defect, and these can build up and even cause cysteine renal calculi or kidney stones. So cysteine, and then the 2 that go with liver problems would be the needle-like one, tyrosine, and the leucine. Whether you say the tree trunk sliced through or little snail, cinnamon roll. But what would you look at too if you're working, you're on evening shift? Before I called leucine and tyrosine, what do you think I'm going to go check? Chemistry panel, liver enzymes. If they have all normal liver enzymes, ALT especially, which is only liver, you better not be calling leucine and tyrosine. You want it to fit. Cholesterol, kind of whatever you think they look like. I think they're like flat pieces of glass with kind of notches in them. And then your sulfa, that's the only drug I'll have you know, but sometimes different drugs you're on can precipitate out in your urine, and the sulfa drugs are a common one. So those are I'm going to pick 12, and you'll do your best 10 out of 12. We'll do it just like last time. And then So that's all in this time. So let's go to the body fluids. This will be for test 5. So the ones we just reviewed were for test 4, yes.
In the lab, do they communicate with each other, like the different departments, like asking like what results they got?
That is your job. You're the tech. You don't have to wait and see if anybody else does it. Like if I'm in heme and I see atypical lymphocytes, what do you think I'm gonna go ask whoever does what test? Atypical lymphocytes? Did you guys not at all relate that to maybe mononucleosis in heme? You're gonna relate it when you do serology. So if I'm working, well, a lot of times I did the serology test too. I would make sure, I'd even take that plasma if I was done on it, spin it down and do the mono test before I reported it out. Because I usually knew they were gonna order the serology mono test. We worked in an area where chemistry was there, heme, UA, CoAg. We were all together except blood bank. And even blood bank, you know, it's different from where it's at now. It's your responsibility to check it out. You have also, anybody remember when Gloria gave her lecture about what I told you my favorite part of having LIS? Remember a thing called a delta check? That you can go back and look at previous results. So you don't even have to go ask anybody. You can go to Mr. Jones for today's labs and see if a mono was even ordered. Or if nothing today, you can go back, hey, this week, he's been in all week, did they ever have a mono test or something? So you're gonna have the LIS to help you too, even if it's, let's say, a Barnes, and you're only working in heme, you can still get on the LIS, look up to see if it relates. But really, that's on you guys. You wanna make sure you're turning out a knowledgeable result and see if it fits. So you'll have access to stuff. LIS is wonderful for a tech because it helps you see what they were running previously. So we'll just go through the spinal fluid, and these are going to be cells that'll be for next week. You're going to have your typical neutrophils, but this is what they would look on a blood smear. This is how they look on a cytospin. So you're going to have to, it puts them all in a clump, and this is a very not cellular spinal fluid. If there's only 5 cells total in one field. You're going to have some of our diffs, you can see in one field and you can almost count your 100 cells in one field, they're so packed. That term pleocytosis. So these all have at least 3 lobes that you can see. So I would report in that field 5 neutrophils. And you don't distinguish, you just say yes, it's a neutrophil. You don't say, oh, if it's a band or a seg. But remember, this is blood, but now you got to relate it to a cytospin smear, okay? And it's off a fluid that sometimes doesn't have a lot of blood. So neutrophils are common. Lymphocytes, this again is your blood, and then this is your cytospin. So what would you report? How many of what on that cytospin on field 1?
7 limbs, 2 neutrals.
So you have 8 lymphocytes and 1 neutrophil. So you're clicking away on your counters and you keep going. You know how you move in fields? Here you got to figure out where you've been to move to the next field so you're not double counting. And then you click away till you get to 100. Many times your predominant cells are going to be the neutrophil and lymph and maybe a few monos on your typical. And usually they're very fast because of the cytospin. If you had to make a wedge smear on a fluid, you could be here all day, it feels like, trying to find enough cells. But the cytospin, all of you should get to see that too. The monocytes, I didn't have a cytospin monocyte one, but you're lucky if the monocytes have vacuoles. But remember, monocytes that don't have vacuoles and large lymphs are easily confused for everybody, even if you're an experienced tech. So, tell me what you remember about a lymph nucleus versus a mononucleus.
It's more circular than nucleus.
What about the density too? Dense or not dense?
They're a little bit more dense than the.
Yep. So it's usually a single nucleus that's good shaped and more compact. What did someone say about the monocyte? Someone back there? Someone say a monocyte nucleus in shape? Someone say indented. A lot of times they're indented. Sometimes they might even be a horseshoe shape. Or they won't always have a nice round nucleus like a lymph. And tell me about the denseness compared to a lymph. Denser. Usually they're softer, the nucleus on a mono. Remember, you need to know your characteristics before you ever look in the scope. So this will be a review too. Sometimes these cells are going to be squished together though, because you're not doing a nice mono layer where they're all sitting nicely without scrunching up. Sometimes the cytospin scrunches them up if it's a cellular fluid. So you got to know your characteristics. Tell me then if it's going to be an immature cell. We already talked about it in relation to renal epis. What are you going to see? Nucleoli. I loved seeing, blasts that had nucleoli. What about the size of the nucleus versus cytoplasm? High NC ratio, kind of like we talked about the one type of renals. And it's a very soft chromatin. You should never call a small lymph, oh, that's a blast for sure, when it has that dense, clumpy chromatin. That's one of the features of a blast, okay? So for me, you're going to have to say it's a malignant or immature cell. Most of the time, these are going to go to a pathologist to be reviewed. You're not going to have to say, oh, yes, that's a leukemia or whatever. But you should be able to at least aware that, hey, that doesn't look like a normal blood cell. And then you won't see many eosinophils, but if there's anything with that allergic reaction, or let's say a fungus or a parasite, you might get more eos. So they typically look like they're. The other thing I always notice, this is a typical looking, a lot of times the eosinophils were two-lobed, and usually they are off to the side, and you could see the nucleus and the granules. Basophils are weird cells, and usually you don't see them in your typical body fluid. I always saw it like, oh my gosh, that looks like a weird cell. And a lot of times basophil, you can't always see the nucleus. And sometimes the granules get washed out. So depending on your staining process, you can't say, oh yeah, they have those big dark granules. Not every basophil. But for some reason, I felt a lot of basophils, the nucleus wasn't as clear cut. Eosinophils, you see, it's usually a bilobed, it's off to the side, and it stands out. Not so in a basophil. So those would be our two that you can really distinctly look for granules. But you shouldn't see really any basophils in your normal body fluids. And I won't worry about plasma cells yet. On lining cells, well, let's go back to that. Tell me what you would distinguish on a plasma cell that we haven't talked about on any other cell. Anybody remember from hematology a plasma cell? What's a key characteristic?
Isn't there slight clearing with cytoplasm opposed to the nucleus?
It's called a paranuclear zone. Did you ever talk about that? This is a distinct feature of a plasma cell, the paranuclear zone. So if you had a multiple myeloma or a macroglobulinemia, you might have a plasma cell present. So you should at least be able to know what is the difference of a plasma cell, and that paranuclear zone is a key feature that kind of makes it stand out. In serology, you're going to learn, anybody know what a plasma cell produces?
Multiple myeloma. A tumor.
What?
A tumor. Multiple myeloma.
No, what does it produce normally in your body that's good? And who becomes it?
Antibodies. It's
It
B
produces
cell.
antibodies, but anybody know what cell becomes a plasma cell?
Lympho B cell.
A lymphocyte type B. You're going to learn about T lymphocytes, helper and cytotoxic, and you're going to learn about B lymphocytes that become plasma cells that produce antibodies so that you protect yourself, okay? So plasma cell dyscrasias, the multiple myeloma, macroglobulinemia, you're going to see on this protein electrophoresis, it's the gamma region that proliferates, and it involves one antibody line being too much. So there's lots of interconnections between a lot of different departments. remember in the lining, these, if I show you a lining cell on test 5 on spinal fluid, if you call it a mesothelial cell, it's wrong. You got to know the name of it from the fluid it comes from. They're called choroid plexus. Another name you'll hear is ependymal. I don't know if I've ever called a lining cell in my whole career on the spinal fluid, but you still got to know it for your registry. I don't think you're going to see them commonly. You're typically just going to see your blood cells, but you got to know what they're called and how to identify them for the registry, and maybe you'll luck out. These I just took. I mean, this is a blast-like cells, but these are all different lining cells. This is probably your best picture. You see the size of a red cell, what white cell is the size of a red cell? What typical white cell? Your small lymphocyte. See how big that is? If a small lymphocyte was the size of that red cell, and it looks like a flat fried egg, but it's purple. Flat, spread out with a big not center nucleus, big nucleus that's nice and round, almost like a lymphocyte. Those are lining cells. So in spinal fluid, you got to tell me that's a choroid plexus or ependymal. If it's a serous fluid, you're going to have to tell me that's a mesothelial. You got to know the names where they're from. But they look the same no matter what fluid they're from. Let me see if I have that blown up at all. If this was a spinal fluid, these are 4, a lot of times they come off in sheets, these lining cells. And this is a small lymph way up here. These would be 4, if this was in spinal fluid, they'd be 4 choroid plexus or ependymal. They look the same no matter what fluid, you just call them different names. But keep in mind, they do not have a 3-dimensional appearance, and they're usually uniform. They look very similar. Blast cells usually have a 3-dimensional appearance, and they all aren't uniform. One doesn't always look like the next. So these are smaller lining cells, but these are your typical lymphocytes. That's how big they are compared to a red cell and a lymphocyte. So keep in mind, before we ever get to look at them for real, mesos are very big cells, probably 2 to 3 times the size of a monocyte. Okay?
So on a test, you'll tell us if it's, what fluid it's in?
Oh yes. You'll know, and next week's test is next week's test will be both of them. So, malignant cells, yes, did you have a question?
The lining cells, those are due to a brain fluid, you said?
Nope, it can just be from the tap, doing the puncture. Doesn't have to be from a bleed. Lining cells aren't anything bad for you. They might just be present because of the procedure. Yeah, lining cells, think of them almost like squamous epithelial cells. They can slough off the area. Think of them, transitional. They're nothing bad. They don't mean anything. So these are some examples. I don't know if you can see the nucleoli at all, but do these cells all look alike? You got big ones, small ones, deeper chromatin, lighter chromatin. That, I would want you to say that's a malignancy or an immature cell. That did not look like the fried egg with a lot of cytoplasm. Look how big the nucleus is compared to the amount of cytoplasm. Did you see on the lining cells, there were probably 3 to 4 times cytoplasm to the nucleus. Know your characteristics and you make a good judgment, okay?
That one on the left, is that like a typical stain or a typical microscope that you would?
These 2 are much better stained. These are 2 deeper. In the third stain, that's the bluish stain. It was maybe too long in it. But these are all typical little lymphs. And look how big, and they're not uniform. And look how big the nucleus is to the cytoplasm. So even not knowing but that one characteristic, you would not call these lining cells. This would be the closest to a lining cell, but it's even half and half. The nucleus is the similar size. So remember, for me, as long as you can tell me it is immature or malignant, That's what I'm gonna expect. Okay? So these are the 3 things for a subarachnoid hemorrhage. This is a macrophage ingesting the red cells. They call it an erythrophage. So I would take macrophage ingesting red cells, I would take erythrophage, but when you have a subarachnoid hemorrhage with a bleed in the brain, you tend to have, what cell becomes a macrophage? You're going to learn this in immunology too. Monocyte in the tissues. It's a macrophage. I think of them as little Pac-Man. Neutrophils and macrophages are your Pac-Man. Then this is the siderophage that has the iron. These are macrophages that have iron precipitate from the breakdown of hemoglobin, which fits. You have a subarachnoid hemorrhage, the red cells are going to break down. So I don't care if you call it hemosiderin, I don't care if you call it a siderophage, but it is iron within the macrophage from the breakdown of red cells. Okay? And then the third thing that you might see on a diff are these, they're called hematoidin crystals. They're little gold bodies. Sometimes this one looks a little reddish, but they're little rhomboid shapes. They're almost like little small rectangular shapes, and they're called hematoidin crystals. So this is test 5. You're going to have to know a lining cell, and I'm going to make another body fluid PowerPoint like I made a urine sediment 3. You're going to have to know an immature cell. You're going to have to know the 3 things for subarachnoid hemorrhage. So one is erythrophage, hematoidin crystal, and the siderophage that had the black precipitate, which was the iron. And you're going to have to know your typical white cells. What's a neutrophil? What's a lymphocyte? What's a monocyte? What's an EO? Which those should be really just review. So that's for test 5 through spinal fluid. And then we'll get to so once again, you can look through the body fluid pictures. Let me go. And once again, you can also do Google Images, because that's where I do a lot of mine from when I make the PowerPoints. Some of them I took from the books, but let me go to I also have a body fluid in-service. Once we get through synovial, I got an in-service that I did way back in the day on CSF, serous, and synovial. And then I made, our disc went bad. It was a little CD cassette. The cassette tape and it all got bad the more and more I used it. So I made slides to fit the in-service. So this body fluid handout side, once we get through synovial, we're going to go through this together. There's 40 slides on it. And then under pictures, you have the body fluid cells. And I put other pictures that I got. Ty from Highland was really good to give me some of his CAP stuff. So there's other pictures from the CAP book, from the urinary cast, urine crystals, under pictures as well. Okay, and then I am going to have another body fluid PowerPoint up by the end of the week as well, before you ever take a body fluid cell test. So remember, tomorrow what we'll review, we'll do lab, and then tomorrow we'll review the aminoacidureas, because that's on test 4 as well. So I can't remember what, tomorrow I think I'm 8 to 11, so I think we can only be in here an hour. So we're going to do our lecture 8 to 9, and then we're going to do lab from 9 to 11, and then you'll break for lunch 11 to 11:30, and then have chemistry. So that's how we'll work tomorrow. We'll start in lectures. I think the person comes in at 9 that lectures on Tuesdays. So